hard week for all of us. We are now plenty of knowledge about viroids. I think it is a privilege for your students and also for me to enjoy the viroid celebrities telling us about this nice pathogen. So far, honestly, I don't know what else can I add to this at this point, but at least I want to invite you to take a walk on the plant side, to move the focus from the viroid. Everybody's in this field, they are thinking of the viroid, for sure. But I would like you to think of the pathogen, to think of the plant. Behind the viroid, there is a plant. So we need to know how the plant is responding to this kind of infection. So my goal now is to give you a general overview of the plant defenses so that you can think of this part of the interaction. So whenever a plant is sensing a biotic stress, it activates the plant defenses so that it becomes, in general terms, more resistant to the future infections. So the idea is to copy the nature's wisdom to design strategies for crop protection, not only by discovering compounds or small RNAs that can be treated the plants to confer the resistance, but also developing more resistant plants. Before continuing, I will just to tell you that I'm going to ask Tanya to upload these presentations for you, for the students, so that you don't have to be taking all the notes, so you will have this for you. So the idea is to copy the wisdom of the nature to design the strategies to have the better results. And for that purpose, we need to research which are these defense mechanisms. So to this, I have designed my talk to cover different general aspects, okay, for the plant defense responses, not only for virus, but the general ones. So in the first part of the talk, I would just review what are the biotic agents causing the activation of the plant defenses, then what is called the systemic resistance, and which kind of systemic resistance has been described in the plants, those activated by insects or the wounding, and those activated by the biotrophic pathogens, including also virus and viroids. And then I will have just an overview, general overview of the silent mechanisms. I know you know about them, but just to refresh and to put everything together in the same fold in your brain, plant defenses. And then I will just finish giving some tips of our own experience in viral immunity. Okay, so starting for the biotic agents, from an agronomic point of view, we can distinguish between which are diseases caused by pathogens and what are pests caused by insects or nematodes. So focusing on the first part of the diseases, we can distinguish between fungi, bacteria, also viruses and viroids. And you have here some examples just for you to remind some of them. For a fungi, those are the classical model systems to study the plant defenses: Cladosporium, Botrytis, in case of bacteria, Pseudomonas syringae, which is also a pathogen we work in the lab with. And viruses, we've seen lots of viruses and viruses in these presentations. So each pathogen has their own nature and their own mode of action. And on the other side, we've got the pests, nematodes and insects, which also have the ability to trigger in the plant specific plant defense responses. So depending on the threat, the plant is going to respond in a way or in another. So, and the way the plant responds after this challenge is first it recognizes the challenge by triggering a local response in that place. But this local response is also accompanied by a systemic response in the leaf that they have not sensed directly the attack. So it means that there are some signals that has to travel along the plant to signal in the defense in the places where the pathogen or the insect is not yet. So I think it is interesting to know about all these mechanisms, and they are specific for the different challenges. For example, for biotrophic pathogens, including viruses and viroids, so the signal molecule has been described to be the salicylic acid, and the systemic response is called the systemic acquired resistance. For the herbivore insects or bunding, the classical molecule, signal molecule, has been described as the jasmonic acid, and the response is called the Boone-induced response or resistance. And there is also a third resistance mechanism, which is activated by beneficial rhizobacteria, which is not so well studied, but is somehow related with jasmonic acid, ethylene, and the activation of the induced systemic resistance. So each kind of mechanism is independent. They are also crosstalk between both of them, but mainly we can summarize that when an insect is attacking the plant, it is triggering the defense against this kind of insects or other. When the pathogen is also infected the plants, is triggering the defense not only for these pathogens, but by this group of pathogens, not against insects. And the same is occurring for the case of the induced systemic resistance. So I have chosen some classic experiments, for example, to demonstrate the Boone-induced resistance. You have here an example of a larvae which are grown in a wild-type plant. Those are the weights of the larvae in a wild-type plant, or in a plant, in a mutant, which is altered in the accumulation of jasmonic acid. You see the larvae are very happy to feed from these plants. So the jasmonic acid for sure is important for the plant defense against these insects. Here you've got an example for the systemic acquired resistance. Those are the local leaves infected with TMV, and this here you can see the symptoms of a second infection in a distal leaf. So you can see that if the SAR is activated, the damage provoked by the virus is more. And finally, you've got the classic also induced systemic resistance, where in this case, they are using Pseudomonas fluorescens in this upper part of the slide, and this is the wild type without trigger the plant with any beneficial insect. So you see, beneficial bacteria. So you see the second challenge is much less aggressive. So it is important to know that this induced systemic resistance is not acting the same as these two, because this is acting like a priming resistance. It's not an established one. The plant is faster responding to the second challenge. So once we know about the different responses, we are going to go a little bit deep in one by one, just reviewing the most important concepts. I know most of you know, but it's to put everything in the same and to think of the plant side, to look for the solutions against the viroids. So in the case of the Boone-induced resistance, you know it is activated not only by the insect, but also by mechanical bonding. You can do with the forceps in the lab. Most of the defense response is going to be similar, but the other thing which is different from both challenges are the volatile organic compounds that in the presence of the insect, they are specific and they can participate in trophic interactions. But as for a general overview, if you put an insect or you forceps you wound the plant, you have more or less the same response. And you have the local tissue damage recognition, the synthesis of the jasmonic acid, which is the main signal molecule participated in this response. And also the reprogramming of the plant, not only at the local level, but also in the systemic levels. And it is important to know not only the signal molecules participating in this process, but also the marker genes behind the activation of this response. So for the case of the bonding response, the marker genes are different. If we are talking about Solanaceae species, the marker classical genes are the pathogens inhibitors. What are these proteins? They are proteins inhibiting the enzymes of the insects so that the insect is not interested in the plant anymore because it doesn't feed properly. So in Solanaceae plants, when you have a plant which is wounded, you can detect the presence of these proteins inhibitors, okay? But in Arabidopsis, there are not protease inhibitors. The classical marker genes for the jasmonic acid response is the PDF 1.2. I think some conferences they've been talking about that. So we have to take in mind that whenever a plant is having these two markers, it is probably being damaged or there is an insect around. So this is the important take-home message is we have to take into account. And specifically, the insects are triggering the jasmonic acid pathway, and the accumulation of the proteinase inhibitor or the PDF 1.2 is occurring not only in the local damaged leaf, but also in the systemic, activating the wound-induced response. So, but locally there are lots of events happening in that there is a party of signaling in the leaf which is damaged, and most of the local events are more or less similar in between the different plant responses. Always it is starting with the signal perception, and then the membrane depolarization, opening the ion channels, and also the calcium is also present many often in all these signaling processes. Activation of kinases, as Dr. Wang tells us about them. And in the particular case of the activation mediated by insects, we can see the activation of the octadecanoate pathway, which leads to the production of the jasmonic acid. So just to know a little bit more about this pathway, so I would like you to invite of which are the experiments that are required to say that a molecule is considered as a signal. So there are three kinds of experiments, like the physiological ones, the pharmac, and genetic. So just because we are tired, just to discuss, what do you think we need to demonstrate that a molecule is considered a signal for a process? Do you think any experiment, also Lynn in the in the virtual world, if they want to participate? Or what do you think? You are not going to get wrong, just to discuss with you.
What happens if you inhibit it?
Perfect. This is one of the classic approaches. You can inhibit either the production or the signaling by pharmacological means or by genetical means. What else? What do you think it is required to consider a molecule? Is it enough for you? It should be enough, but we can think of extra experiments.
Phenotype, what? Is it obvious? Can you measure it?
So if you can measure, it is supposed to be enhanced upward. Okay, so if you compare two plants, one with the response activated and one with not, the signal should be enhanced in the activated. So it has to be enhanced. You can, if you apply the molecule, it is supposed to activate the process. No? What do you think? So those are the kind of experiments we perform in the lab to demonstrate that the molecule is acting as a defense, as a signal molecule. So just, do you want to add anything?
It needs to be localized to the place.
Yes,
of the injury.
it has to be localized and also travel along to the distal tissues. That's perfect. Anything else you want to add? More or less we have covered all the experiments we should need. So in fact, the jasmonic acid is induced upon wounding, not only in the local, but also in the distal tissues. As you have said, if you apply exogenous treatments, you can see the activation of the marker genes, be the proteinase inhibitor or the PDF1. And also if you prevent the accumulation, even with the pharmacological approach or in the mutants, you can't see the induction of the protein markers anymore. So those are the classical experiments we perform in the lab to look for signal molecules. First, we need to compare wild, not challenge and challenge plants to detect differential molecules, and then we treat the plants with the molecules, and we generate plants with altered levels of the molecules and analyze the plant response in these plants. This is how we work in the lab normally. So for the jasmonic acid, they perform, not my lab, but other labs, they perform that experiment. And this is a classic paper in the plant defense response, because the jasmonate, particularly the methyl ester of the jasmonate, is a volatile compound which can activate the defense not only in the plant, but within the plants. which is the first WhatsApp between the plants, you know, in terms of chemical communication. So when you are bonding a plant, the plant is emitting this aroma, and the plants around are also activated. It's like a social meeting event, okay? So if you use the jasmonic acid as a perfume, you have to be careful because you can be triggering the plants in the lab. So it's because it's a volatile. So there are lots of perfumes containing the jasmonic because it comes from the jasmine flower. So you have to take care of that if you want to your control plants free from the markers. So you can't use the perfume for that. So we are happy to in our lab, we are happy with this paper because we are also working with volatile organic compounds. So you've got the pathway. I don't know if it's worthy. It is not worthy to deep into the chemical, but just to have you a general overview, it is started in the chloroplast by releasing compounds in the membrane because you broke it. So starting from the fatty acid coming from the membrane, and then several conversions that occur in the chloroplast, leading to the production of the OPDA, which is a 12-oxophytodinoic acid, which is also an active molecule, not only the jasmonic acid, but sometimes the intermediate in the pathway are also active. So that is why it is important to invest a little bit of time of having a general overview of the pathway, because not only the final product, but some intermediate products are also active. So this OPDA is coming to the peroxisome, where this sometimes reducing and oxidated several times, and it comes to the cytosol, where it's converted into jasmonic acid, and at least in Arabidopsis, the jasmonic acid is conjugated to the amino acid isoleucine, and this is what is this day considered as the active form, the jasmonic isoleucine form, which is the ultimate signal in the Bund response. So you've got here the different intermediates, and some of them have been described to be also active, not only the jasmonic, but OPDA, the jasmonic isoleucine, also conjugated to tryptophan. So you have to look not only to the final signal molecule, but also the related molecules can also have an important role in the signaling, or they are candidates for looking for crop protection, not only because sometimes it is too expensive, but a different form is more cheap. So for the companies, it's better to use a different compound, not the final. So once we know that the jasmonic acid is the classical signal for the response to wounding an insect, so we need to know also the signaling transduction pathway behind the jasmonic acid signaling. And this is just an example of how to decipher the transduction pathways of the signal molecules, which is by making screenings. So you have to design your own screening. This is a very simple one, because the jasmonic acid treatments in Arabidopsis plant, it causes this reduction of the growth. So this research group, they look for insensitive mutants, which cannot either sense or signal the signal of the jasmonic. So it was a very easy screen, because you just grow the seeds in the presence of jasmonic and see the ones that are insensitive, they can see the jasmonic. So this is the way they identify the classical gene for the jasmonic acid response, which is COI1. It is COI because the name comes from coronatin insensitive 1 mutant. Coronatin is a jasmonic acid analog, which is present in the bacteria. The bacteria, they are so clever, they have this compound to inhibit the defense. I don't have the time to enter, but it's an analog of jasmonic acid. So this group, they identified this mutant, the COI1, and they observe that this mutant is not activating the jasmonic regulated genes anymore. So the plants are hyper susceptible to the insect, but also to the necrotrophic, because these two response are together. And this protein is an F-box acting through the degradation of the 26 proteasome. So after performing different screening, they could decipher the whole pathway for the jasmonic acid, and they find the JAS protein also as a mutant in the screening. And JAS proteins are repressors of the wound response. They are normally acting in impairing the MYC2 to activate the genes. So this is the JAS genes acting on MYC, repressing the induction of the genes. So whenever the jasmonic acid is produced, then this CoI1 is perceiving the jasmonic acid and acting on the JAS genes. So the CoI is destroying the repressor, so de-repressing the response. So normally, in terms of plant defense, the things are normally repressed, and the signal is de-repressing, because it's faster to repress than to look for the promoters. So they are ready to go, 3, 2, 1, and then they repress. Normally, this is a common mechanism by acting, destroying all the JAS, and then in the presence of jasmonic acid, the JAS are destroyed, and then the MYC2 can activate all these proteinase inhibitor, or in the case of solanacea, or PDF1. So this is how the wound response is acting. If you want to ask me anything on that. So it's important to keep in mind the way to identify the molecules, which is by comparing the profiles of the healthy and challenged plants, and then to discover to demonstrate that they are indeed signal molecules, and then to decipher the transduction pathway and to get the final products. So in the same manner, we are going to talk about the systemic acquired resistance that maybe is the most interested for us. It is activated by biotrophic pathogen infection. Those are pathogens that need the plant to be alive to continue the cycle, not the necrotrophic, but the biotrophic. And as we have been telling, the salicylic acid is required for the action, well, for the response to this kind of pathogens. And the gene markers are the pathogenesis-related proteins, the PR proteins. So I'm happy to say that in our group, not myself, but my directors, they participate in the discovering of this kind of PR proteins. And this is the reason why the viroid is a good system to study the plant response, because it's a free protein pathogen. Everything that you see in the plant is because of the plant, not because of the pathogen. The signals, the proteins, whatever. So it was a clean system, the viroid-infected plants, to identify the PR molecules, because the PR pathogenesis-related proteins come from the plant, not from the pathogen. So in our group, we have also a celebrity, which is Vicente Conejero. You have been enjoying lots of celebrity people, viral people, also talking about former celebrity people. So it's for me a pleasure also to talk about my celebrity man, which is Vicente Conejero, who participated in the discovery of these PR proteins in tomato plants. So those are our markers for the infection of biotrophic pathogens. So this kind of systemic acquired resistance, at the beginning it was described related to the floors model, in which we have 4 different scenarios, depending on the presence of the resistant gene or not in the plant, and the presence of the avirulence genes or not in the pathogens. Only in one case, when the R gene is recognizing the ABR1 gene, this is what is called the incompatible interaction. Then the local response is activated in the leaf, and then the systemic acquired resistance is developed. But it used to be like that, but then you have here the example. We work in the lab, tomato Rio Grande plants carrying the PTO gene, the resistant, with the bacteria carrying the ABR PTO. So you see the recognition, and then the activation of the plant response is occurring. But if the bacteria has not the ABR PTO, so the plant can't see the pathogen, and then it can't display the plant defense, and then we are in these possibilities, and then the disease is occurring. So after this classical floors model system, the PTI came to our lives with the zigzag model. And it seems to be a very easy model, but it's very difficult to understand and to explain, at least for me. So the thing is, well, just to make mention to Helen and one, they have been demonstrated in this Congress that the PTI is also occurring for viroids and virus. So for me, it's a very nice news because we were out of the system when we started with the zigzag model with people working on viroids and virus. We were not in this system, but thanks to them, we are in again with the PTI. So just very briefly, the PTI is occurring when it's the first layer defense, and it's occurring when the plant perceives like general molecular patterns, for example, the flagelling of the bacteria. You know, and then this occurs because there are specific receptors for these plant molecular pathogens. And then we have two scenarios. When I'm going to focus on the bacteria system because it's the one who allowed the definition of the zigzag model. So the bacteria, normally it has effectors. So the PTI is not occurring in a normal bacterial infection because those effectors are inhibiting the PTI. So as a normal thing, the effectors are inhibiting the PTI, and you can see the PTI when you infect with the bacteria. It's not a normal response that you can see, at least infecting bacteria. So these effectors, we have the two possibilities. They can be recognized, therefore triggering the ETI, effector trigger immunity, which is the Flores model, or not. They are not recognized, so then the infection progresses. So it's important that, and there are also resistance, it's important that you understand that the bacteria, normally when you infect with the bacteria, you can't see the PTI because the bacteria has the effectors repressing this response. You just can see ETI or not ETI. That's it, the Flores model. So, but when you use bacteria which can inject the effectors, or when you use the flagellin 22 or just boil bacteria, then you can see the PTI, okay? So in a similar model, way of the jasmonic acid, it's been experimented to demonstrate that the salicylic acid is a signal molecule, the same as you have been saying. The salicylic acid are enhanced upon infection. If you treated the plants with salicylic acid, you can activate the defense. And also we've got these plants, the nachi plants, which are unable to accumulate the salicylic acid so that the defense are not activated and they are hypersusceptible. So these days, not only the salicylic acid, but new molecules have been proposed to be the signal molecules for plant defense, such as hydroxypipecolic acid and also azelaic acid, lysotyl-3-phosphate. There are many other molecules, not only the salicylic acid, because it's difficult to understand, because the salicylic acid is necessary. If we have not salicylic acid, the plant defense response is not activated. But when a plant is dying, the higher amount of salicylic acid is produced. So it's not sufficient. It is necessary, but it's not sufficient, the salicylic acid production. So there are two different pathways to produce the salicylic acid, the isochorismate pathway and the phenylalanine pathway. This is the one much related with the pathogen infection, the isochorismate synthase, and And there are two genes here which are classically related with the activation of ICS, which is SART1 and CBT, just for you to be used to hear about them. And I'm very happy to say that in 2025, in July, there have been two different Nature papers discovering the new phenylalanine pathway to the production of salicylic acid, just for you to know. It is not that important for pathogens because in the context of pathogen is the ICS pathway, the important one, but I think it's worth it to mention that there are two different papers at the same journal deciphering this pathway. So the salicylic acid, in the same manner as the jasmonic acid, it has also some derivatives. Some of them are the gentisic acid, some glycosylate. Also, the methyl salicylate is an important molecule because, as the jasmonic acid, the methyl ester of the salicylic acid is a volatile compound. So again, it can communicate to the rest of the plants that something is happening. So it's a volatile compound, just to keep in mind. This is important. And in a similar manner for the COI1, it has been deciphered the pathway of the signaling pathway for the salicylic acid, and Siniandon is the main actress of this decipher And in this case, they perform a very smart screening in order to identify the components. So the screening, it is not the important, the screening per se, is for you to think of possibilities, okay? So she took the promoter of PR1 and he fused to a reporter gene so that if you infect the plant, you can see the blue, okay? And she looked for insensitive response mutants, so mutants that if you infected, you can see the blue. So they are called non-expressor PR1 mutants and PR1 mutants, okay? So that's how she discovered NPR1, which is the main. So NPR1 is 2-salicylic acid as CoI1 to jasmonic acid, okay, just for you. Even that it is not well admitted that is the receptor, because after that they came the NPR3 and NPR4 as receptors for the salicylic acid, and there is like a theory that depending on the amount of salicylic acid, you can detect these three scenarios in which NPR1 is the higher affinity receptor and the NPR3 is the lower affinity, and then you can combine the three possibilities displaying either ATI or SAR. And then the thing is not ended here, because she has now discovered these condensates which are similar to those described by Constantina for BIP1. Those are condensates that are normally accumulated in the cytoplasm, displaying ETI, or accumulated in the nuclear, then displaying SAR. So it's a very complex model. It's even, I read it every time and it's very difficult to understand, but in general terms, you just have to keep in mind the salicylic acid, NPR1, NPR3 and 4, and the formation of the condensates. So far, we've seen that we are not focused on the beneficial rhizobacteria because I think it's not that interesting for you, but so far, we've seen that the wound response is activated jasmonic acid and the wound-induced response, and the biotrophic pathogens are activated the salicylic acid and the systemic acquired resistance. So just to make you put everything together in your mind, so the silent mechanisms has been described like a side way of defending the plant, but it's also a plant defense mechanism. And the origin of this, as you know, of this mechanism comes from the co-suppression lines, where you were overexpressing something and you get the other way around. And these days, we can know that they are the most important component participating in this response are first the double-stranded RNA. They can come artificially or naturally in the plant. The dicer, which are chopping this double-stranded, but this is important to remind that these dicers are inspecific. They chop any double-stranded gene, and then as a result of this cleavage, one of these small derived RNAs is incorporated into the RISC, and this is the specific. It patrols the cell looking for the complementary sequence in it, and then cleavage specifically the target sequence. So those are the three important things to keep in mind. I know they are obvious for you, but just in case, I'd like to remind you this. And the main components are in Arabidopsis. There are several dicers, several agos, and several RDR. And just to have a general overview of this silencing mechanism, I like this classification in between the exogenous activated RNA silencing and the endogenous activated RNA silencing. So in the exogenous, we can talk about the inverted repeated sequences that we introduce in the plant to get the RNA interference plants. Also, the sense post-transcriptional gene silencing, the ones that can explain the co-suppression. When you overexpress whatever, the plant feels that this is not the normal way to accumulate that RNA, that something is happening and the silencing mechanism is activated. And also, for sure, the one activated by viruses in plants and viroids, and also by wounding, and also by pathogens. They have been described several silencing mechanisms for all the pathogens. And then the endogenous, naturally occurring, RNA silencing plants, and we can distinguish in between the microRNAs and the small interfering RNAs, which I don't know if it is updated, because every time I look at it, there is a new small RNA coming in my life that I have to study. I don't know if I am updated anymore, but as far as I know, we found the fuzzy and tassy small RNAs, the natural small RNAs, the heterochromatic small RNAs, the long small RNAs, and maybe a new one in 5 minutes is coming to our life. So pay attention to that, because this community is very productive. So I have prepared the slides to explain all of them, but I think we are tired, so I'm skipping them. Just if you want, you have the information in the presentation in case you need an extra. And to finish, which is our small contribution to this plant defense response for the viroid, I am not investing my time on that because Teodora has made my job with her poster, and he has been explaining all our contributions to discovering compounds that somehow are participating in the plant defense response, and they are, I think, effective against viroids. So they need to be checked in field conditions, but we have some examples such as, for sure, the salicylic acid, GABA, ethylene, or some TOR inhibitors that could be good candidates for Teodora to check our system as a proof of concept. And we hope we could discover new ones that could give you solutions for the crop protection. So the takeaway messages I would like to tell you is that there is a systemic resistance which is activated specifically for each pathogen or group of pathogens. I haven't tell you about that, but it's like if we sleep, we can attend the lesson at first hour. So if we are defending against the pathogens, we can't be defending against the insect. There is a crosstalk between the two pathways. So the plant, if you trigger the salicylic acid pathway, then the plant will be more resistant to biotrophic, but more susceptible to insects, and the other way around, because resources are limited. If we go for a drink, we are now a little bit tired because our resources are also. And the future research questions, in my case, in my personal opinion, I would like you to think of how can we leverage the systemic resistance to get sustainable crop protection. We can inspire ourselves in the nature, because I think it is much more clever that we can be, because the evolution is smart. So try to copy as much as we can the mechanisms. And also, I think is, for me, also a very important question. I was talking also with Teodora in the breakfast, because these things are to share meals. And so if, as far as I know, we can't so far uncouple the symptoms from the defense, at least for the viroid infection. So we don't know if the symptoms are the plant trying to defend to the viroid or the other way around. We don't know which is the feature of the viroids which make them to cause symptoms. So for me, this is the main question that we would like to solve. I mean, we've seen that there are some viroids living in the plants without giving symptomatology, and some of them with the same structure are not causing severe symptoms. So for me, the main question that we should all together work in, also to, apart from trying to develop a crop protection, is to understand how the viroids are causing diseases in the plants. So I invite you to think with me of this, and I for sure will be available for you. Whatever you need from me, I will be more than pleased. It's for me a pleasure to be part of this community, because I'm like young in this community, because my expertise, not young, but young in this community. I'm just 3 Congress or 2 or 3 Congress. So if you have any doubt, we can discuss whatever you want. Thank you very much.